Welcome to the course on basic genetics. The teachers here are Dr. Richa Arya, Professor Madhu Tapadia, and myself, Jagat Kumar Roy. We are from Department of Zoology. This presentation is of week 10, first lecture, and the title is Karyotyping. Human chromosomes, as observed under the microscope, looks like these. So this is a plate which is photographed straight from the microscope. This is a normal bright field microscopy image. Now we want to have human chromosomes. So which tissue should be suitable? The skin, is that suitable? Can we take a piece of skin? Answer is no. Can we take a piece of liver? Answer is no. Lung? Answer is no. Muscle? Answer is no. Can we take bone marrow, which is very good tissue for cell division studies? We cannot take, because these are invasive techniques where one has to remove a piece of tissue, and that is neither ethically correct nor one is allowed to do that. So blood, that is of great choice, because that we can draw from the body. In the blood, we have the RBC, we have WBCs, we have platelets. Which cell type? The RBCs are enucleated, so they will not have chromosomes. WBCs and platelets, they have the nuclei. Platelets are small cells. The WBCs, they have the good nuclei, so some of them can be of choice. Among the WBC, the lymphocytes are specifically important, because they can be made to divide if we want to. Otherwise, in the blood, they are not dividing. But if they interact with antigen, let us imagine some kind of infection comes, and if they interact with the infection, then they start dividing, making more number of cells and producing antibodies and so on. So we have to set, as I said, that the lymphocytes are the cells of interest for making the chromosomes. They have to made to divide. If they have to divide, that means we have to place them in culture. So for setting lymphocyte culture, 5 ml of blood is drawn from the body in a sterile heparinized syringe in suitably clean environment. Heparinized because this will prevent blood clotting. Otherwise, clotted blood will have lots of lymphocytes that will be gone with the clot, and they will not be of use for us. For culture, some of the following things are needed. Number 1, tissue culture medium. 5 ml of tissue culture medium can be taken. Then fetal bovine serum. We do not know what are the other factors. Most of the factors, those are required for cell division. Few we know, few we do not know. In the blood serum, most of those factors are there which will allow cell division to occur. So that is why fetal bovine serum we use. Serum means the blood minus the cellular parts. So the tissue culture medium plus serum, that constituted the medium. In that, we have to add 0.3 ml of blood. Another thing we need is phytohemagglutinin. What is phytohemagglutinin? This serves as an antigen, and this is from plant source. This serves as an antigen. So this triggers the lymphocyte to divide. That is why we are placing phytohemagglutinin 0.1 ml. And the pH is also important. If the pH is alkaline, the cells will be killed. So what you do, you blow carbon dioxide in the culture. How you can do? You can use the carbon dioxide cylinder and you can bubble through that, or else the simplest and oldest technique is pipette should be closed from the mouth side with cotton plug and one can blow air through that pipette. So whatever infectious things are there in the mouth, that will be kept there, that will not be allowed to go in the blow, and only carbon dioxide that we are blowing, that will enter in the culture, and that will keep the pH of the culture okay. The culture is kept in an incubator at 37 degrees centigrade for 48 to 72 hours. Why 48 to 72 hours? Because after triggering human cell, it undergoes one cycle in approximately 20-24 hour time. So if you are keeping for 48 hours, 2 cycles of cell division will be there. 72 hours, 3 cycles. So 2 to 3 cycles of cell division will be there. That means we will have plenty of cells which are in different stages of cell division. Then culture must be inspected every month because we are keeping culture for 3 days. There should not be any infection. So one has to watch, and also pH has to be checked. It is not that pH is being checked with pH paper or pH meter. There are indicators present in the medium. So by seeing the color of the medium, one can guess what kind of pH the medium is having. And accordingly, little bit of carbon dioxide may be required to be blown so that pH becomes okay. Then about 2 to 3 hours prior to harvesting the culture, harvesting means prior to terminating the culture or taking out the cells from culture for chromosome preparation. So for this, we add colchicine or colcimate. Both are the plant product, and they stop the spindle fiber assembly. That means microtubule polymerization is inhibited. Why are we putting this? Because we want metaphase chromosomes. If a cell is in metaphase, this will pass to anaphase, but if colchicine is present, this cannot go to anaphase, this will remain at metaphase because spindle fibers will not be made, so that the chromosome pulling will not be, the chromatids will not be pulled to opposite poles. Now next is chromosome slide preparation and its staining. So what one does, that the culture is transferred to a centrifuge tube, because now our aim is to collect these cells. So to collect the cells, we have to centrifuge so that cells are collected in the form of pellet and the medium remains in the form of supernatant. So at 1000 to 1200 RPM for 5 minutes, this is okay for allowing the cells to get pelleted. The supernatant is discarded or decanted and cell pellet is resuspended. So resuspension means once again we shake the cells so that it is not a clump, and this is resuspended in pre-warmed means 37 degrees centigrade hypotonic solution. What is hypotonic solution? This is 0.56% KCl Why do we call it as hypotonic? Because this has less salt concentration than our body fluid. So, this will cause the swelling of the cell. Once the cell come to suspension, the volume is maintained to 8 to 10 ml and incubated at 37 degrees centigrade for 20 minutes. This will allow the cells to swell. What is maximum swelling? The maximum swelling is that a cell is not bursted, it is maximally swollen. So, in metaphase, the nuclear membrane is not there. So, when hypotonic treatment is given, the chromosomes which are otherwise close by, they get little bit spaced, and that will help in having a better metaphase chromosome preparation and identification of chromosome. So, that is why hypotonic is given. Many a time one says that too much hypotonic treatment may allow the cells to burst. As such, the cells do not get bursted, they leak and they tend to keep a swollen condition. So, 25 minutes or so, 20 to 25 minutes time is okay as hypotonic treatment. Once again, we have to centrifuge cells at 1000 or 1200 RPM for 5 minutes. This is again to collect the cells. This is to collect the cells which have been treated with hypotonic solution. The hypotonic to be decanted, and now the cells are to be fixed. What do we mean by fixation? The cell should be remaining in its same state, in its same condition. What fixation does? This removes water, this will cross-link the other molecule, so the situation will remain and will not get deteriorated or will not get rotten after a period of time. What we will use as a fixative? The good fixative is 1:3 acetic acid methanol. This fixative is good for nuclei, good for chromosome fixation. So, after the hypotonic is decanted, drop by drop gradually, one should not add the fixative, gradually drop by drop, one adds the fixative and start agitating so that there is no clump formed. The cells are suspended. Many add a few drops of fixative at the end of hypotonic treatment itself. This is a slow fixation so that there is no shock. So, the fixative is added, that also made 8 to 10 ml and left at room temperature for 15 minutes. Always one change of fixative is very good, because when we fixed first time after hypotonic, some of the hypotonic drops remained in the tube, and once we fixed, it is not that aqueous solution is totally gone, some of the aqueous part remaining with the fixative. If we do second time fixation, water will be totally gone and the fixation will be better, the morphology will be better. So, one can fix once again, or if time does not permit, one can centrifuge, take this, collect the cells, decant the supernatant and resuspend the pellet in fresh fixative, only 0.2 to 0.4 ml. This is for making chromosome preparation and keep at room temperature for 10 minutes or so. Then we have to make the slide. So, a clean slide is taken. With the help of a pasture pipette, one draws some amount of cell suspension and allows it to fall on the slide from a distance, say from 10-inch distance or 12-inch distance. This jerk by which the cells fall on the slide will make the spreading better. So, that is why from a distance one thinks that this helps. Then immediately after putting the cells on the slide, allow the slide to get burned on a flame. It is not heating, it is just showing the slide to a flame. The slide will immediately catch fire because a lot of methanol is there in the fixative, and then the flame will go. That's it. So this way the cells are dried, the slide is dried. At the same time, this helps in charring the cytoplasm. So now the cytoplasmic material is charred, and once whenever you wash the slide, the cytoplasm will go. Nuclear material, nothing happens to it. This will be on the slide, and thus this will make a good preparation. This will give you a good preparation. Chromosomes are usually stained with Giemsa. So Giemsa is a very good stain. Stain for 5 to 10 minutes with 5% Giemsa. That is the standard one. And then rinse twice in distilled water, or even tap water is okay. Distilled water with pH near 7 is better. Otherwise, tap water is fine. Dry the slide completely and mount with a cover glass. So your slide is now ready for observation. As shown in the first picture, I am showing the same picture again. This is human chromosome plate. Now a photograph is taken. After taking the photograph, these chromosomes are cut with the help of scissors like this, one by one. These chromosomes are cut. And then they are allowed, all the chromosomes are to be cut, and then they are paired. So we have to make karyotype. Karyotype means the chromosome should be organized in a fashion so that by seeing the karyotype, one is able to see whether the karyotype is normal, whether this has some abnormality, and so on. So the chromosomes cutouts are arranged pairwise in the descending order of size. Large chromosomes first, the smaller chromosomes later, like that, in that orientation. So this is size as well as considering the shape. The shapes of the chromosomes are metacentric, submetacentric, and acrocentric, the three shapes. So these chromosomes are pairwise put on the paper and stuck to it. What we will obtain, I'll show you. Just for a minute, the shape of chromosomes. You all know this is a chromosome. This is a chromosome. You all know. This is the centromeric region. The centromeric region is having constriction. With protein, this looks maybe a little bit thicker, but otherwise this is DNA wise, this is a constricted region. This is present in all the chromosomes, so this is called as primary constriction. In a chromosome, there can be another constriction in some of the chromosomes, not in all the chromosomes. If such constrictions are there, like this region, this is known as secondary constriction. Not this. This is secondary constriction. So some of the chromosomes contain secondary constriction. This part, that means the part of the chromosome, the main body is here. This is the main body that is here. This part appears separated from main body. And that is why this is called as satellite. Satellites are those which is separated from main body and still has a relation. So here this appears separated from the main body, but not actually separated. They are attached with a constriction. So these satellite region or this constricted region, this is very important for a cell, because here the This is the nucleolar organizing region. This is called as NOR, secondary constriction as NOR, or nucleolar organizing region. This is nucleolar organizing region. And thus they are called as NOR. How they will organize nucleolus? The nucleolus region is having ribosomal RNA. So, the chromosome with ribosomal RNA genes, this region, this has repetitive ribosomal RNA genes. So, these regions remain decondensed, and that is why appear condensed, and they make profuse amount of ribosomal RNA. When too much of ribosomal RNA is made, that region, the nearby region with RNA with some of the protein which binds with those RNAs, they make a cloud which appears different from the other part of the nucleus. So, this is within the nucleus. All those chromosomes which have the satellite part, or all those chromosomes which has the nucleolar organizing region, if they are nearby and if they are making lot of ribosomal RNA, a ribosomal RNA cloud will be around the chromosomes, around that region. And thus, this will appear as the nucleolus. Maybe I will try to show you with the diagram also. Okay. Then the other things, this is a metacentric chromosome. Metacentric means the centromere is in the middle. This arm, one arm, you call it as P arm, the other arm you call it as Q arm. So, this is Q, this is P. If P arm is equal to Q, then you call it as metacentric chromosome. Submetacentric chromosome when P arm is shorter than Q arm. So, P is shorter than Q arm. Then this is the submetacentric chromosome. Then the other chromosome is the having very little P arm or almost non-existent and long Q arm. So, that is your acrocentric chromosome. This is, so when the chromosomes are arranged, it appears like this. So, the chromosome pair 1, 2, 3, 4, 5, 6 like this, all the 46 chromosomes are arranged pairwise, and this is the human karyotype. If we describe the human karyotype, how did we keep the chromosomes? The 3 pairs, then 2 pairs, then 6 pairs, then 3 pairs, 3 pairs, 2 pairs, 3 pairs, and so on. So, how this is done? If I see in this slide that chromosomes are organized in 7 groups. Group A containing chromosome 1 to 3, these are large metacentrics. Group B, chromosome 4 and 5, large submetacentrics. Group C, chromosome 6 to 12, and as well as X chromosome, they are median submetacentrics. Group D, chromosome 13 to 15, this is medium sized acrocentric with satellite. Group E, 16 to 18, small metacentrics. Group F, chromosome 19 and 20, small metacentric. The group E, I said small metacentric, this is small submetacentric. And group G, chromosome 21, 22 and Y, these are smallest acrocentrics with satellite. Okay. So, with this we come to the discussion. I will suggest that take a printout of human metaphase plate and prepare a karyotype by yourself. Why is phytohemagglutinin used in lymphocyte culture? I did talk, but you should once again have a discussion. Why did we use colchicine? hypotonic and fixative for making chromosome preparations? And why did you flame dry after putting the cell suspension on slide? I briefly described all this, but you can have once again revisit and once again understand this thing. Thank you so much.